Hello, and welcome back to Introduction to Genetics and Evolution. In the previous set of videos, we talked about testing for whether populations fit the assumptions of the Hardy-Weinberg equilibrium. At the very end of the last set of videos, we looked particularly at one deviation from the Hardy-Weinberg equilibrium, that of the Volland effect. That is what happens when you sample two populations simultaneously that differ in some of their allele frequencies, and as a result of that, you see an underrepresentation of heterozygotes. Let's recap that for just a moment, and then we'll move into how we can actually leverage this Volland effect for actually quantifying differences between populations in allele frequency. Now, what we did last time is we tried to calculate Hardy-Weinberg expectations and fit the Hardy-Weinberg expectations for this MN blood type. Now, I showed you data from Navajo and our Aborigines. Here's the data from the Navajo. So we first have our raw genotype counts. From that, we got the total number of individuals. Using these two together, we then were able to calculate the observed genotype frequencies. From the observed genotype frequencies, we calculate out the observed allele frequencies here. And then from these observed allele frequencies, we look at the expected or predicted Hardy-Weinberg genotype frequencies, and these are down here. In this particular case, with the Navajo alone, we see a pretty good fit between the observed genotype frequencies and the predicted genotype frequencies. Now, what happens when we sample across two populations simultaneously? Well, if we sample from Navajo and Aborigine together, we follow the same overall procedure, but in this case, now when we look at the predicted genotype frequencies, they do not very well match the observed genotype frequencies. In particular, we see this deficiency of heterozygotes in the observed relative to the predicted. At 0.488, the predicted number is much greater than the observed 0.246. I attributed this to the Volland effect. That is what happens when you have populations that differ in allele frequencies. Even if each population individually is at Hardy-Weinberg, if you put them together, you see a deficit of heterozygotes. Now, how is it that populations differ? Well, I like to look at this in two ways. One possible is they may have different allele and genotype frequencies. That is what we've been emphasizing so far. We'll come back to that in just a moment. But first, I wanted to entertain another possibility. There may be alleles at some genes that are found in some populations, but not found in other populations. You may have some alleles that are found, for example, just in East Asians, but you never see those particular alleles at other populations. Now, the genes would be there, but the particular alleles would not. This is especially likely under two particular conditions. One is if it's a very recent new mutation that arose in an East Asian population, for example, and hasn't spread to others. Alternatively, if the populations are in complete isolation, then that difference could persist for quite some time. So let's talk just briefly about this latter group, and then we'll come back to the former. Now, differences can arise by mutation and spread. So let's say in an ancestral population, everybody was little a, little b, little b. You can have over time, let's say for example, the population splits. So this is indicating a split between the populations. So in one population here, the one on the right, you have a mutation from little b to big B. This big B allele can spread, and eventually everybody here may be little a, big B, big B. In this other population, the one on the left, you can have a mutation from little a to big A. The big A allele can spread, and eventually everybody in this population can be big A, little b, little b. So again, we have in this case a split between populations, and we have differences that can then arise and potentially spread. This is especially likely if you have complete isolation. We'll come back to this actually when we talk about speciation later on. Now, groups within species are different, yet they are related. That they may have some of these unique alleles that are not found in other groups, but it's probably more likely that most of them have very different genotype or allele frequencies instead. This shows approximate relationships from one calculation of various human ethnic groups. So, for example, Melanesian and Papuan are more closely related to each other than either is two populations from the Mediterranean or from Siberia, etc. Now, how do we quantify these differences? Well, the easiest thing to quantify is if everybody in one population differs from everybody else in another population at some allele. This is referred to as a fixed difference, right? So, for example, in population 1, everybody's big A, population 2, everybody's little a, little a. So we're looking specifically at the A gene in this case. If everybody in one population differs from everybody in the other population, we refer to this as a fixed difference. If you go back to the ancestor, presumably they had the same alleles, but something arose, at least in one population, and spread to make it so it's completely different from the ancestor and from the others. Now, this does happen, but it's not very common within a species, and it's generally not true among modern human ethnic groups. Instead, as I said, it's more common that you have frequency differences of alleles and genotypes. For example, big A may have a frequency of 0.7 in East Asians, but 0.5 in Indian populations. That's just as an example. Now, what we're trying to do here, let me emphasize this, is we're measuring differences between populations, not individuals. We cannot say just because somebody has big A, oh, therefore they are of Middle Eastern descent or something like that. It's based on the overall population. The relative abundance of particular alleles differs in one population as a whole relative to other populations as a whole. Well, this poses a challenge. How do we quantify these slight differences in frequency between populations? Well, the deviation from Hardy-Weinberg that's associated with the Volland effect that I've already introduced actually allows you to quantify allele frequency differences between populations. So let's assume that two populations are at Hardy-Weinberg, and that is an important assumption. We'll come back to that later. So if you sample each population by itself, you'll see them at Hardy-Weinberg. So this would be similar to what we saw with the Aborigines and the Navajo example I showed you earlier. But if you sample both together, we see this deviation from Hardy-Weinberg. And again, as you saw, that was the Volland effect. Now, how big this deviation is from Hardy-Weinberg when sampling these populations simultaneously will quantify the difference in allele frequencies. That if you're fairly close, then the allele frequencies must be similar. If you're very different, then you'll see a very large Volland effect. So the measure that we'll use is referred to as FST. So it's F with subscript ST. Now this measure ranges from 0 to 1. If you have 0, then there's absolutely no allele frequency differences between the populations you're studying. If it's between 0 and 1, then the allele frequencies differ somewhat, and if it's 1, you actually have this fixed sequence difference, such that, for example, everybody in this population is big A, everybody in that population is little a. They don't actually have to all be the same. Maybe all of these would be A1, or A1A2, or A2A2, and over here in this other population, they're all A3A3, or A3A4, or A4A4. Basically, it just means you have no overlap in alleles between this population and that population. Now the measure is very simple. So this FST can be estimated as the Hardy-Weinberg predicted 2PQ, basically what would happen if you were at Hardy-Weinberg, minus the percent observed heterozygotes divided by Hardy-Weinberg predicted 2PQ. Essentially, it's the predicted minus the observed over the predicted, which is fairly common. Let me show you an application of this, and you'll see this is very straightforward. So let's say that there's an example, as I mentioned to you, of fixed difference. Population 1, everybody's big A. Population 2, everybody's little a. So here's the totals. Now let's imagine we're sampling these two populations together, and we have to assume that we're sampling similar numbers here to calculate FST correctly. So here's our pooled population sample, 100, 0, 100. Well, if we calculate this out, the total genotype count is 200. When we look at the genotype frequencies, the observed would be 0.5, 0, and 0.5, right? Half the individuals we're sampling from this pooled population are big A, half from are little a, none of them are big A. Here are our allele frequencies, 0.5 and 0.5, right? All straightforward so far. Our Hardy-Weinberg predicted 2PQ should be 0.5. And what we see is this contrasts dramatically from our observed. So here's our Hardy-Weinberg predicted, here's our observed, and we see a striking contrast between these two. Now we apply that formula I just gave you. This is the Hardy-Weinberg predicted 0.5 minus the observed 0 divided by 0.5, and this gives you an FST of 1. As I mentioned, if you have an FST of 1, that indicates a fixed difference. Let's try one that's not quite so extreme. Here's a set of populations. Each population size here is 1,000, so we have our total population size is 2,000. This population is at Hardy-Weinberg. This population is also at Hardy-Weinberg, but they differ in allele frequencies. So here's our pooled population. I just summed these numbers together. 250 plus 90 is 340. 250 plus 490 is 740, etc. So again, what we do is we calculate the totals, get the genotype frequencies, get the allele frequencies. So here's our total, 2,000. Here's our genotype frequencies, which is each of those numbers divided by 2,000. Here's our allele frequencies. It actually comes out very nicely to 0.6 and 0.4. So what is our Hardy-Weinberg predicted 2PQ? Well, it'd be 2 times 0.6 times 0.4. So 0.6 times 4 is 0.24. So our predicted should be 0.48. My math in my head is correct. That is what we see. In contrast, the observed is 0.46. So again, we have a slight difference here. It's not a huge difference in this case, but it is noticeable nonetheless. Well, now we can calculate a much smaller FST than we did before. Here's our number. Here's our Hardy-Weinberg predicted, 0.48, minus 0.46, which is the observed, divided by 0.48. Again, it's always the predicted minus the observed over the predicted. In this case, our FST measure comes out to 0.042. And again, in this case, we have small differences in allele frequencies. The previous example, we had a very large difference in allele frequencies. And you can see that FST ranges from 0 to 1, 0 being no difference, 1 being complete difference. Let me give you one to try. Here is the mixed data from the Aborigines and Navajo. The numbers here aren't perfect because the sample sizes weren't exactly right, but let's just pretend for a moment that they are. Go ahead and calculate what you would see as FST in this example for Aborigine to Navajo. Well, I hope that wasn't too difficult. Let's go ahead and run through them. Again, just to remind you what FST is. FST is the Hardy-Weinberg predicted minus the observed heterozygotes over the predicted. So let's put in the numbers. Here's our allele frequencies. Here's our expected or Hardy-Weinberg predicted. We do see a deviation in this case. Here's our predicted is 0.488. Our observed is 0.246. So what should FST be? Well, FST would be in this case 0.488 minus 0.246 over 0.488. Comes out not exactly, but approximately 0.5. So in this case, we see a fairly large FST, and you'd expect this, that there's been no historical gene flow between Aborigine and Navajo. Now again, to recap, FST is larger when you're comparing populations that are more different in allele frequencies. So again, the Aborigine and Navajo are very different allele frequencies, and that's why we saw a fairly large FST there of 0.5. If the frequencies were identical, FST would be 0. If they were fixed different, FST would be 1. Now let me give you a couple of values so you get an idea of what we actually see in human populations. Obviously, Aborigine and Navajo is an extreme example. Among human populations, this is from a 2010 study that used a little bit over 1 million SNPs. If you look at African Americans relative to Europeans, FST was about 0.11, so that's noticeable but not tremendous. African Americans to Chinese, FST was 0.15. Europeans to Chinese, FST was about 0.11. And if you look among European populations, let's say, for example, you compare the Spanish to the Italians to the Germans, things like that, FST is typically, not always, but typically less than 0.01. It's very, very low as you look at that sort of scale. Let me show you a big table. There's a big table of FST measures. I'm not going to walk through all of these, but you can see that there are some cases which are fairly high. Like over here, we have a couple that are over 0.2, like POP1 to Red Sea, etc. Several of these are high, several of these are low, but you see this range of values. Now, what is FST in words? Well, we can define FST as the percent heterozygous of randomly chosen alleles within populations, that's the observed aspect, relative to what would be expected in the entire population. Okay? It's the percent heterozygous of randomly chosen alleles within populations relative to what would be expected if it was completely randomly interbreeding. And again, it's measuring this difference in allele frequencies. So why don't we see higher FST among human populations? That we know there's a lot of non-random mating out there. Why isn't it bigger? Well, there's a couple of reasons. First, some of the assumptions of FST are violated in humans, that this is supposed to be applied to genes experiencing little or no natural selection. You recall I mentioned earlier that FST should be applied when each of the individual populations is known to be at Hardy-Weinberg. Now, some of the SNPs, for example, that were studied in that previous example may not be neutral. In fact, some of that variation may be under some sort of selection. It's also susceptible to differences and historic changes in population size among groups. But probably the biggest reason we don't see higher FST values is because we actually do have a fair amount of gene flow, or closer to random mating among population. That'll be the subject for the next video. Thank you.